cancer of colon, lung, and larynx. As we all know that selenium is a very important trace element which is required by our body for the proper metabolic functioning, and its concentration in the body has a U-shaped curve relationship with respect to all-cause mortality and cancer incidence. The selenium acts in our body through two main known mechanisms, and the one is neutralization and detoxification of heavy metals like arsenic, cadmium, nickel, chromium, beryllium, lead, and mercury. And second, through incorporation of selenocysteine to form selenoproteins. And there are 25 known selenoproteins in the human genome, and they are responsible for various functions like oxidative stress, thyroid hormone regulation, immune response, apoptosis, cell division and proliferation, phospholipid synthesis. And selenium has been associated with many diseases like Keshan disease, Kashin-Bach disease, Hashimoto's thyroiditis, Graves' disease, asthma, HIV, neurological disorders like Alzheimer's, Parkinson, epilepsy, schizophrenia, and type 2 diabetes, and cancer, which is my area of research. Recent studies have indicated that selected macroelements and vitamins can cause, they have significant influence on the cancer risk, and both deficiency in excess can cause dysfunction for specific proteins which can lead to cancer. CHEK2 is a cell cycle gene which is involved in DNA repair pathway, and mutation in CHEK2 gene may cause cancer. And 6% of the Polish population are the carriers of one of the 4 founder mutations of CHEK2 mutation. And these mutations are associated with varied risk of different cancers like breast, prostate, thyroid, kidney, and colon cancer. And this variability in the risk of cancer can be explained by genetic and environmental factors. And in our study, we have considered selenoprotein coding genes as the genetic modifier and concentration of selenium in the body as an environmental factor. So this is a very interesting question which came up during my research. Is CHEK2 a selenoprotein? The answer is no. Or we can say it's not yet known. Because the formation of selenoprotein requires specific components like selenocysteine insertion sequence, which is abbreviated as SESIS, and UGA codon, which is normally works as a stop codon, and it is present in the 3' end of the mRNA. And interestingly, CHEK2 has all these special components, but there is no scientific evidence of insertion of selenocysteine through the SESIS pathway. Also, certain functions of CHEK2 are very similar to selenoproteins, which indicates that there may be some kind of interaction, or they may belong to the same class of protein family. And the aim of my study was to analyze the influence of selenium concentration and variation in selenoprotein coding genes, which can be used as a marker for cancer detection in Polish population with CHEK2 mutation carriers and unselected lung, laryngeal, and colorectal cancer. Material and methods. During our study, we have collected 156 cancer patients with CHEK2 mutations and 612 healthy controls. These case controls are matched in 1:2 or 1:1 ratio. And in another group of unselected cancers, we have collected 274 unselected patients of lung, laryngeal, and colorectal, and also 274 healthy controls. And these case controls were matched in 1:1 ratio. And the criteria for case control matching was sex of a patient, year of birth, cancer family history among the first degree relatives, smoking, pack per year, and CHEK2 mutation type only for the CHEK2 group. And we performed the measurement of selenium through ICP-MS, and we used a strong quality control criteria to avoid any bias between measurement of cases and controls. We used reference material measured every 4 samples, and cases and controls were tested consecutively or alternately. And the measurement accuracy was plus minus 5%. For genotyping, we performed genotyping in 4 common variations in the cellular protein genes, namely GPX1, GPX4, TXNRD2, SEP15. And we used real-time PCR analysis by using Techman probes on light cycler. And estimation of cancer incident analysis was performed under the influence of selenium level and or genotypes of the cellular protein genes. For selenium level analysis, it was performed in categories and quartiles, and the categories were less than 60, 60 to 70, 70 to 80, and more than 80 microgram per liter. And we tested 4 variations. So for each variation, there were 3 genotypes for the tested gene. And we also studied the combined effect of selenium level and cellular protein genotypes, and to see the mutual effect in cancer incidence. And for statistical analysis, we used conditional logistic regression for the matched case control study, and multiple testing methods like Buffer correction was used only for genotype analysis, and they were all performed in R. So first I will discuss the results of check 2 carriers. We can see in this table that the mean selenium level in all cancer cases of check 2 carriers are lower, except ovarian cancer. However, the mean selenium level difference between cases and controls are only significant in all check 2 carriers, in prostate cancer and kidney cancer. And the cancer incidence in check 2 carriers, it increases, it decreases with the higher selenium level. We can see here in the quartile analysis for check 2 carriers, we saw decrease of more than 2 times in the cancer incidence with selenium level greater than 86.28 microgram per liter, and in the category analysis, we see a significant decrease of more than 1.5 times with selenium level greater than 80 microgram per liter. And in the check 2 subgroup analysis, the elementary cancer incidence was significantly decreased with to almost more than 6 times with selenium level greater than 83.92 microgram per liter. And but the selenium level analysis was not significantly associated with the other subgroups of check 2, like breast, prostate, lung, and the renal cancer. And the analysis of cellular protein genotypes, we saw that the carriers of AA genotype of the selected variant in TXNRD2 gene, it shows more than 2.5 times increased cancer risk with and without the influence of selenium. And similarly, the AA genotype of SEP15 gene, and the variant is RS5845, it also shows more than 2.5 times increase of cancer incident under the influence of selenium. And these p-values are all non-formally corrected. And in the subgroup analysis of check 2, we saw that the carriers of AA genotype in the SEP15 variant, it shows approximately more than 5 times the increase of cancer incidence of breast cancer. And also, the CT genotype of the selected variant in GPX1, it shows more than 6 times increase of cancer incidence with a very significant p-value. And now I'll list the result of unselected cancers. Here we can see a very significant difference between cases and controls in the unselected group. So all the unselected cancers are very strongly difference in case and control, the mean selenium level. And we saw that lung cancer incidence, it decreases with the higher selenium level. And our study shows that with the increase of selenium level of greater than 79.98 in the quartile analysis, it shows 10 times decrease of cancer of the lung cancer incidence. And similarly in the category analysis, it shows 10 times decrease. Here quartile and category are quite comparable, that's why results are, the odds ratios are quite same. And in the laryngeal cancer incidence, it also shows a decreasing trend with increase of selenium level. And here also we see in the quartile level analysis, the laryngeal cancer incidence was decreased almost 7 times with the selenium level of greater than 85.52 microgram per liter. And in the categorical analysis, it shows more than 4 times of decrease of cancer of laryngeal cancer incidence. And colorectal cancer also reveals a similar finding. With increase of higher selenium level, there was decrease in colorectal cancer incidence. And for quartile level analysis, it shows more than 7 times decrease of colorectal cancer incidence with selenium level greater than 81.66. And for categorical analysis, it shows more than 4 times decrease in cancer incidence. And the genotype analysis shows the carriers of CT genotype of the selected variant in GPX1 gene shows more than 3 times reduction in cancer incidence under the influence of selenium. And the genotypes of the other selected cellular protein coding genes were not significantly associated with laryngeal and colorectal cancer incidence. And to conclude my results, higher selenium concentration is significantly associated with lower probability of cancer incidence in CHEK2 carriers and unselected cancer patients with lung, laryngeal and colorectal. And selenium concentration can be used as a valuable marker for early detection of cancer in group of CHEK2 carriers and unselected patients with lung, laryngeal and colorectal. And the effect of selenium level in blood serum on cancer incidence may depend on the genotypes in cellular protein genes. And at the end, I would like to acknowledge people who are responsible for accomplishment of my PhD work. First of all, I would like to thank Professor Anna Jakubowska, my supervisor, and Professor Jan Lubinsky, the head of the department, for my scientific, technical, and personal support all these years. And I would also like to thank my colleagues, Kasia, Jaworska, Marcin, Kasia Katmarek, Kasia Durda, Gregor, Magdalena, Anirudh, Tamara, Thierry, and all my other lab mates. And I'm also grateful to Mrs. Eva, who was always ready to help me in any official work. And Anna Waidas is my flatmate. She was helping me in collecting my literature data during my PhD writing. And I would also like to thank all the staff members from the Department of Genetics and Pathology. And I would like to thank my reviewers, Professor Andrzej Polski and Professor Bogdan sorry. And last but not the least, I would like to thank my friends from Poland and India and my sweet family in India waiting desperately for me. Thanks a lot.
Thank you, Satish. So now I would like to introduce Professor Bogdan Gałbrzewski, first reviewer from the Medical University of Łódź, also the Department of Genetics in Medical University of Łódź. So we would like to hear your view.
Ja przygotowałem swoją recenzję w języku angielskim, tak kolega będzie miał pytania, chętnie się do nich ustosunkuję, a fragmenty recenzji przeczytam w języku polskim. Chcę podziękować panu dziekanowi za zaproszenie mnie do recenzowania rozprawy doktorskiej kolegi Satisha Gupty. Monografia złożona na moje ręce, licząca 103 strony maszynopisu, posiada typowy dla dysertacji doktorskich układ treści. W rozdziale pierwszym, liczącym 10 stron, opisano historię naturalną tego pierwiastka, zwracając szczególną uwagę na dane dotyczące metabolizmu oraz źródeł pozyskiwania tego pierwiastka. W rozdziałach kolejnych opisano metabolizm różnych postaci selenoprotein, aby w rozdziałach 4 do 6 skoncentrować się na roli selenu w chorobie nowotworowej. Istnieją dane literaturowe pokazujące istnienie związku pomiędzy poziomem selenu w krwi obwodowej a ryzykiem rozwoju chorób nowotworowych Badania dowodzą, że uzupełnianie diety selenem może mieć zastosowanie w efektywnej profilaktyce nowotworów. Wyniki pokazują również, że zmiany w obrębie genów selenoprotein mogą mieć związek z ryzykiem wystąpienia choroby nowotworowej. Powyższe fakty skłoniły kolegę Satisha Guptę do podjęcia badań naukowych nad wyższymi zależnościami w reprezentatywnej grupie osób obarczonych mutacją CHEK2 oraz osób nie będących nosicielami wyżej wymienionej mutacji. W dodatkowo prezentowanym projekcie oznaczano stężenie selenu w surowicy w grupie nosicieli mutacji CHEK2 oraz nieselekcjonowanych pacjentów z rakiem płuc, krtani, jelita grubego, a także u osób zdrowych. W tej grupie chorych badano 4 różne polimorfizmy, które kolega zaprezentował. Pomiary stężenia selenu w surowicy u osób z badanych grup wykonano z użyciem metody spektrometrii masowej, natomiast analizę wybranych w genach selenoprotein wykonano przy pomocy tej techniki real-time PCR. Wyniki analiz pokazały, że wysokie stężenie selenu w surowicy wiąże się z zmniejszoną częstością występowania raków u nosicieli mutacji CHEK oraz raków płuc, krtani, jelita grubego niezwiązanych z mutacją CHEK2. Wydaje się również, że polimorfizmy w genach kodujących selenoproteiny, takie jak wymienione przez kolegę, mogą mieć związek z ryzykiem raku u nosicieli mutacji CHEK2. Podsumowując, w oparciu o powyższe dane oraz dane literaturowe autor stwierdza, że niskie stężenie selenu w surowicy może służyć jako marker umożliwiający selekcję osób do diagnostycznych badań przesiewowych, takich jak tomografia komputerowa umożliwiająca diagnostykę raka płuc i oskrzeli, czy też kolonoskopia służąca diagnostyce raka jelita grubego. Ponadto autor sugeruje włączenie oceny stężenia selenu do programu badań kontrolnych u nosicieli mutacji CHEK2. Myślę, że wykorzystanie poziomu selenu jako markera do selekcji osób kwalifikowanych do tomografii i kolonoskopii natrafi na opór ortodoksyjnego środowiska klinicystów w naszym kraju, co nie musi oznaczać, że kolega Gupta nie ma racji. Istotnym walorem badań prezentowanych przez kolegę jest reprezentatywna liczba osób i pacjentów, która została objęta badaniami, znacząca liczba osób, które stanowiły układ odniesienia oraz metoda oznaczania poziomu selenu. Zarówno materiał badawczy, jak i metoda badawcza są unikalnymi w skali kraju i znaczącymi w Europie. Domyślam się, że kolega Satish Gupta mógł mieć ograniczony udział w kreowaniu warunków do przeprowadzenia badań, ale być może nadchodzi również w naszym kraju taki moment, że osoba pragnąca przeprowadzić określony projekt badawczy nie musi rozpoczynać wszystkiego od zera. Kolega Gupta skorzystał z możliwości, które zastał i trafnie z nich skorzystał. W literaturze światowej toczy się ożywiona naukowa dyskusja związana z rolą selenu w etiopatogenecie choroby nowotworowej. Ta dyskusja nie ominęła naszego kraju, gdzie problem ten wzbudza bardzo silne emocje. Kolega Gupta włączył się do tej dyskusji w sposób merytoryczny i znaczący. Tym głosem dyskusji jest nie tylko monografia będąca dysertacją doktorską jego autorstwa, ale przede wszystkim publikacje, które ukazały się w czasopismach z listy filadelfijskiej w liczbie 3. Biorąc pod uwagę przytoczone wyżej okoliczności, stawiam wniosek do Wysokiej Rady o dopuszczenie kandydata do dalszych etapów przewodu doktorskiego. Stawiam również wniosek do Wysokiej Rady o wyróżnienie tej rozprawy. Dziękuję.
I teraz poprosiłbym profesora, ponieważ profesor Pławski, recenzent, nie mógł wyjechać, prosiłbym profesora Węgnaka o przeczytanie głównych części jego recenzji.
Doctorate thesis of Mr. Satish Gupta entitled Influence of Selenium Concentration and Variations in Selenium protein genes on the CHEK-dependent cancers and unselected cancers of the colon, lung, and larynx was performed at the Department of Genetics and Pathology of Lenin Medical University under the supervision of Professor Anna Kowalska. The dissertation consists of 103 single-sided printed pages and covers introduction, aim of study, material methods, result, discussion, conclusions, abstract. Results obtained during realization of the above thesis have been published in 3 international journals. Therefore, I reckon the doctorate meets the requirements of the Obrony Medical University in Szczecin for the possible application to the world. Neoplastic diseases are one of the most important medical, social, and economic problems in Poland and around the world. Cancer, after cardiovascular disease, is the major cause of death in Poland. The incidence of malignant neoplasms reached over 140,000 in 2010, of which about 70,000 are males and 75,000 are females. There are several high-risk genes predisposing to cancers. The mutations in the BRCA1 and BRCA2 genes are strongly associated with breast and ovarian cancer risk. About 10% of colon cancers are associated with hereditary with HNPCC syndrome and FIP, caused by mutations in DNA repair genes. Mutations of the retinoblastoma 1 gene cause retinoblastoma and hyperlindal disease, VHL disease. Most cancers, which are not related to strong genetic predispositions, become a challenge for today's researchers. The low and moderate risk genes play an important role in predisposition to cancer initiation and or development. Still, the cancer susceptibility conditioned by low and moderate risk genes has to be modified by other factors. In cancer prophylaxis, regardless of the genetic background, the early detection of cancer is crucial for the effectiveness of treatment. The search for markers allowing early detection of cancer is the challenge for modern science. The search for such factors has been the subject of research of Professor Levinsky's team for many years. Research issues of Master of Science Satish Gupta thesis fits into the general trend of the team's studies. The aim of the study was clearly defined and was to determine whether the selenium level in serum and or alterations in selenoproteins or the genes can be used as a marker for cancer detection in Polish CHEK2 mutation carriers and in persons with unselected cancers of the lung, laryngeal, and colorectal. To check this hypothesis, the selenium concentration in serum was determined in 3 groups, followed by the analysis of 4 variations in selenoprotein genes, GPX1, 4, PXNR-B2, and SEP15. The selenium concentration in entire imaged CHEK2 carriers was significantly lower in cancer patients than in healthy controls. The probability of cancer detection was over 2 times lower for individuals with selenium above 86 micrograms per liter in comparison to the individuals with selenium below 65 micrograms per liter. The selenium concentration was lowest in patients with cancers of respiratory tract and prostate, while highest in breast and ovary cancers. The prostate and lung laryngeal cancer incidence was not significantly associated with selenium level in the blood serum. The studies of selenium concentration in the group with unselected cancers show similar correlations as in entire matched CHEK2 carriers, but the significantly lower selenium concentration was observed in all types of cancers in this group. Analysis of alterations in selenoprotein genes shows that they found significant association of AA genotype in RS11397 and RS5845 with increased cancer risk in all CHEK2 mutation carriers. In different cancer groups, significant association of AA genotype in RS5845 with almost 5 times increased risk of breast cancer was observed. The association of CT genotype in RS10504 with over 6 times increased prostate cancer risk, it was statistically significant. The results are very interesting and indicate a significant reduction of serum selenium concentration in cancer patients, both checkpoint mutation carriers and unselected ones. Master of Science Satish Gupta undertook very difficult and complex studies on the search for cancer marker in a group of cancer patients, including checkpoint gene mutation carriers and unselected patients. The low selenium level in serum was associated with several-fold higher incidence of cancer, particularly in the group of unselected tumors. The strongest association of low serum selenium concentration was observed in the case of lung and laryngeal cancers. Serum selenium concentration seems to be a valuable marker for selection of individuals for surveillance examinations such as CT for detection of the earliest stages of the bronchial cancers or colonoscopy for early diagnosis of colorectal cancer. The results significantly expanded our knowledge and understanding about the role of selenium in cancers. The impact of these findings have been confirmed by publications of obtained results in 3 articles in international journals with impact factor 3, 3.7, and 3.5. The manuscript was prepared very professionally in hardcover. Figures and the tables are well described, clearly and logical complement the text of doctoral dissertation. In the introduction, PhD student presented the knowledge concerning selenium, its metabolism and malignant diseases in the context of the doctoral dissertation. The sentence, the mutations in APC gene was reported in 35% of the Polish families with FIP, is based on the oldest publication considered this issue and is not up to date. I found the selection of methods correct. The test group is well described by the PhD student. The results are submitted in the form of summary, which in case of such extensive material is preferred. In summary, the discussed above PhD thesis is of a very good quality. Therefore, I am applying to the Scientific Council of the Faculty of Medicine, Biotechnology and Laboratory Medicine from Rhine Medical University in Szczecin to grant an award summa cum laude to Master of Science Satish Gupta.
Thank you. So Satish, would you like to respond for reviewer remarks, suggestions?
There is one comment about I have put the whole data about the mutation of APC gene in Polish, the percent of mutation of APC gene in Polish population with fat disorder. I have put 35%, but the latest data says 54%. In the paper of Professor Andrzej Płaski, he has analyzed 300 probands, and in that he found 166 probands with 97 different kind of mutation. Of that, 80 are small mutations and 17 are large rearrangements. So this updated information is 54% of the APC gene mutation in Polish population with FAP.
Any remark for Professor Kałczewski? Any response?
There is no.
You can always thank.
Thanks.
So, would you like to ask questions for this dissertation? Discussion, sorry.
Maybe just comment on extension of this review performed by Professor Kołczewski. We are really lucky that we have a chance to invite to ask Professor Kołczewski to review our thesis, because this is he is one of a few persons who really understand what we are doing. And your comments were already matching the focus of this studies, of merit of the studies. Just one small extension about your comment concerning the contribution of Dr. Gupta, or Satish Gupta, we are before voting, so of Satish Gupta by definition, all of these works which we are doing, we really are going to be serious and really value the contribution is no, practically, there is no chance to achieve something working just alone. And here you have a classical situation. The same, let's say, pairs, cases, controls are used by different people, and everything is elaborated by few people, right? The same with microelement analysis. It's not just one person who is able and who is supposed seriously to do something. But, however, we are expecting all the times that PhD students will have their own special contribution. And in this case, the situation is extremely clear. There's a part, for example, concerning analysis of CHEK2 like potential cellular protein, and it is done completely by himself. I was not, I'm, let's say, a kind of consultant for these studies. I was not aware of his activity. I know about the role which promoter was playing in these studies. But certainly, this is his personal contribution to his studies. There is no doubt that this is just by him. So, and this is what we should expect in my understanding of this, what we are doing, what we are getting when studies performed within a group of people, what is a necessary way of achieving some results. But thank you for these comments.
Your success started several years ago from the registry of patients and the bank of DNA. The beginning, there is the biggest value. Of course, you know, ideas and so on, but registry is step number one.
I know that of patients, it is recommended that selenium was supplemented in some patients or in a group of patients. What do you think about selenium supplementation? Does it change the incidence of cancer in this group of patients who are on selenium, and for how long they should be on selenium? I don't know, years, so I have no idea, but I know it is, this study is probably done, maybe there are some results already available about that. And from, because one of your conclusion is that certain level of selenium is a recommendation for further studies like CT for lung cancer. Yes. So what level of selenium and how it is changing on supplementation? How high it should be to improve the outcome?
Based on my study, if a selenium concentration is more than 80 micrograms per liter, you have a less chance of getting a cancer. I'm not talking about the risk, but the chance of getting a cancer, or probability or incidence of cancer. So, because this is a retrospective study, so we don't know whether these levels of selenium in the body or in the blood serum is a cause of cancer or is a result of cancer. So this is a retrospective study, so we can't say much. But based on our result, we can say, like, okay, if we supplement population, especially polyps population, where the selenium level of polyps population is 40 to 50, and if you are supplementing 55 micrograms per day, daily, and it reaches to more than 80 micrograms per liter, you have better chance of kind of survival, you can say survival or protection against few of the mechanisms which are causing cancer. But there are many prospective studies and there are many clinical trials which says that if you have a selenium concentration higher than some baseline, there are 2 studies, clinical trials, which say that if you have more than 135 milligram per microliter of selenium, you have less chance or less risk of having a cancer. So we can use as a marker for detection of cancer in polyps population, as my conclusion says.
Should it be lifelong supplementation?
It won't be lifelong. Like if you reach a certain level, and like in Finland, the selenium concentration was quite low in the Finnish population. Then they introduced selenium in the fertilizers, and through food chain it's reaching to the normal population. And they attain this selenium, which is the optimal for that population, and they're getting benefited of this optimal concentration. So when you know that you have reached optimal concentration, I think it should not be recommended because it leads to selenium toxicity.
I have a question. And what is selenium, medium selenium level in India?
In India, it is 28 to 40.
So,
But I was reading few papers, and it is not very clear because there is not much study. Because it's a huge country, and it depends, like which region you are studying. It's like broadly speaking, if you say northern region or the southern region. But there are few papers which say it's like 28 to 40 micrograms.
so it's quite low level.
Yes.
Because my question is because in Pakistan, yes, so also it is different. So I saw the paper, so the medium level was about 200.
Actually, this one which I'm talking, it's about like it's a daily intake. But there are few papers which talks about esophageal cancer or the cancers of alimentary tract. There they have studied. They found that there is a lower selenium level in cases compared to the controls, but it was like not significant. But compared to the controls, healthy controls, it was lower, and they say it's like around 120. And personally, my selenium level is 90 microgram per day.
Any other questions?
Just one comment. I think that there's a huge problem of underestimation of the difficulties in getting proper result about selenium level. I can tell you clearly and honestly that at the beginning our ambition was not to measure selenium level. We thought about ourselves that we are specialists on DNA analysis, not on selenium measurement, and we sent samples to several laboratories, obviously in COVID form. So for control, we sent the same sample with different codes, and the results were like disaster. Completely unreliable. So when I see the difference between India and Pakistan from 20 to 200, I would start by checking reliability of laboratory analysis.
Any other questions? So if not, so thank you very much. So the next step is so I think we will thank you very much.
Thank you very much.
And now we can make our job.
It was a pleasure, no job.
Thank you very much.